So today topic of CellTalk presentation is organ aging signatures in the plasma proteome, track health and disease. It will be presented by Dr. Nupur Biswas. Nupur Biswas is working as a senior bioinformatics scientist at Winix. She is working for bioinformatics and other multiple projects as well. Some of her projects include designing personalized new antigen vaccines for cancer patients and exploring the genes behind chordoma and the health applications of digital healthcare wearables. Nupur has a doctoral degree in physics and her research work focused on studies of confined polymers. Before joining Unix, she worked at the Indian Institute of Science, Rama Research Institute, Collab and the Indian Institute of Chemical Biology. At Unix, she has gained experience as not only a research mentor but as a scientist contributing to various international publications. Now, request Dr. Nupur to start the presentation.
Thanks, Shreya, for your nice introduction. Welcome, everybody. Today, I am going to present a paper which is titled Organ Aging Signatures in the Plasma Proteome Tract Health and Disease. It was published in the journal Nature, which has an impact of 64.8, and it was published in the year 2023. This research is primarily focused in the longevity, but it has impact also on neurodegenerative diseases, cognitive behavior, and cardiac diseases. So today's talk is about aging. Aging is actually time-dependent deterioration of the functions of human organs, which affects our health. So understanding aging is important because it affects both at the population level and also at the individual level. Well, it affects the massive disease burden of the aging because aging is accompanied with the several chronic diseases. So if we can understand this aging in a better way, it can revolutionize patient care, preventive medicine, and drug development. But onset and effects of aging varies across individuals, and the susceptibility and resilience to age-related diseases in specific organs such as brain, heart, kidney, which are considered as the major organs of our body, that also varies substantially across the population. Now, as we age, our cells undergo several biochemical changes, and this actually leads to organ damage. It increases risk of chronic diseases, and finally, it leads to death. We know that aging varies between individuals, but at the same time, animal studies also show that aging varies between organs of an individual also. So in this context, the challenge is that there are many methods to measure molecular aging, but they provide a single measure of aging for the whole body. Specific information specific to the organ aging is a challenge. There are clinical chemistry which provides organ-specific markers, but they have low organ specificities. And few methods are there which are organ-specific, but they are expensive and they lack molecular insights. In this context, objective of the current research article is stated that how human organs change molecularly with age. Do all organs age at some rate and then measure with aging across the body? To address this question, the researchers followed this methodology, which started with identifying organ-enriched genes. Organ-enriched genes means the genes which are more expressed in the specific organs. And then they mapped the plasma proteins to the putative organ resources. Then they used machine learning algorithms for different cohorts and calculated individual organ age gaps and associated these organ age gaps with organ aging phenotypes. To tell the methodology about details, they actually considered 5 cohorts, which comprised more than 5,000 subjects and almost 5,000 plasma proteins. They mapped these proteins to putative organ sources. And they considered genes as organ enriched only if they are expressed at 4 times higher in any specific organ compared to any other organ. In this way, they identified more than 800 organ-enriched proteins. And then they used these organ-enriched proteins to model organ aging. They introduced a concept, age gap. This age gap measured an individual's biological age relative to other same-aged peers based on their molecular profile. And then they associate this age gap with the mortality risk and age-related other phenotypes. In the machine learning context, they use class aging models for 11 major organs using mutually exclusive organ-enriched proteins, trained organismal model using, these are the names of the models. They have given organismal model, conventional models, this using different types of proteins, organ non-specific proteins, organ-specific proteins, and training cohort had 1,398 healthy participants, having mean age 75, which means they are considering mostly elderly population. And they tested it on 4 independent cohorts. Their results show that individuals with the same conventional age gap, like this brown gray circle, which is close to 0 for each individual, 1, 2, 3, these 3 are 3 individuals, 3 example individuals, which results are there showing. Although the conventional age gap is similar, but the organ-specific age gap varies a lot. You see, for this ID2, pancreas is almost -2 age gap, and heart is almost close to 1.5. These are the data from 3 randomly chosen individuals. When it is studied in the population level, that is to the whole cohort, this resulted in a low to moderate correlation between the age gaps of different organs. They also noticed that some individuals have extreme aging in one particular organ. Like these 3 individuals they are showing, these 3 circles are showing very high organ age gap. That means for these individuals, some organs are aging more fast. Such individuals are considered as outliers if they have any given organ with a very high age gap. And these extreme persons are considered as the extreme aging types. They further did some correlation analysis. They did the aggregation into distinct organ etiologies. And they found that aging in one organ actually did not occur with aging in other organ for such people. And this type of extremely aging people actually comprise 23% of the whole cohort. But among them, only 1.7% individuals showed extreme aging in multiple organs sometimes simultaneously. They also have studied association between organ aging with the biological aging, as the biological aging comes with many age-related diseases. We consider few diseases like hypertension. Individuals with hypertension had kidneys that were approximately 1 year older than their same age peers. Same age peers means which has not a diseases. And individuals with diabetes had kidneys approximately 1.3 years older, and individuals with atrial fibrillation had a heart 2.8 years older. Now they focused on the kidney aging related proteins which are highly expressed in the kidney cells. They identified the proteins which are related to the kidney aging and found these are the proteins and they are all expressed in different types of kidney cells like proximal tubule, podocytes, connective tubule. And among them, they identified REN protein as the most important kidney aging protein. They further did a longitudinal follow-up study for the 15 years data, spanning 15 years data. In this 15 years follow-up study, they observed for people having no active disease or clinical, at least no expressed disease, having no clinical abnormal biomarkers, every 4 years of additional heart age caused 22.5-fold increased risk of heart failure. Similar trend was observed for other organs also, but not so much high. For kidney, the risk is only 1.5-fold around. They further associated brain aging with neurodegenerative diseases. They focused on neurodegenerative diseases because they usually comes with the aging, and they are very common for the aged people. They identified the brain proteins contributed to the association between the age gap and the cognitive decline, and these proteins are shown here. If here API-BFP is an algorithm that was developed by them, and then they associated this with the chronological age and found there are clusters of proteins also who are associated with brain aging with the neurodegenerative diseases. And a total of 47 out of these 49 model proteins were actually detectable in the human brain RNA-seq data, and they are mostly mapped to the neurons. Either neurons, and few of them were also found at the oligodendrocytes. Now they looked at how aging of other organs contribute to the brain aging phenotypes. They used 4 aging models for cognitively healthy cohort. They observed that the decreased cognition function was significantly associated with all 4 age gaps. They used correlated artery, pancreas with the brain. Then has found the association between age gaps and the risk of transition from cognitively normal to mild cognitive impairment. That means how with being aged, a cognitively healthy person can become a person with cognitive impairment over the 15 years follow-up period. And they found that cognitive organizational and cognitive artery age gap significantly can predict the occurrence of the impairment. So, for them, hazard ratios are quite high. And then they are studied the biological processes and proteins involved in early cognitive decline. They plotted the aging trajectory of the model proteins. Here, they observed that cognitive organism and cognitive artery proteins, which they identified earlier, they changed with age earlier and at a faster rate. And cognition brain and cognition pancreas protein actually changed quite slower rate. So if this study introduces a framework of modeling organ health and biological using plasma proteomics, the resulting organ aging models can predict mortality, organ-specific functional decline, disease risk, and progression, and aging heterogeneity between tissues. This minimal invasive approach, because this is very much less invasive because it needs only the blood sample, it can be applied to further understand the effects of health intervention such as lifestyle modifications, drug therapies at the organ level. Future studies are needed to assess how proteomic organ aging relates to other molecular measures of aging and disease, such as methylation aging clocks and disease-specific prediction models. Now, many proteins of this study have genetic associations with diseases of their respective organs, and they are actually validated therapeutic targets. And this leads to a potential causal role of these proteins in organ aging. Future studies are also needed for exploring the genetic architecture of the organ aging clocks, the relationship of clocks to disease using GWAS and post-GWAS methods like co-localization and Mendelian randomization, considering larger cohort. The limitation of the study includes it considers a subset of organs. Here, the cohort is mainly comprised of the older adults that we already mentioned. The average age is 75 years, and it was comprised of mostly American and Caucasian people. So, other ethnicity like Asian cohorts can also be studied. In conclusion, this study shows that large-scale plasma proteomics and machine learning can be leveraged to non-invasively measure organ health and aging in living people. If they use sets of organ-specific proteins to get subset of physiological age-related proteins, it shows different rates of aging within an individual and measurement of aging at organ-level resolutions. The impact of the study is that it developed a methodology for tracking organ aging using plasma proteomics data. That's all for today. Thanks you everybody for your attention. If you have further questions, any comments, you can write to me at nupur@genix.org.
So there is a question from Vijay. Was extreme aging of any organ attributed to lifestyle or other dietary causes? How were the factors other than genetic factors controlled for?
Okay, yeah, these questions are really important, but the current study did not address other than the genomic effects, genomic context. This study did not address did not include the effect of dietary intakes in the study. They just explored whether the plasma proteomics data can be used for the organ aging. This study actually more on developing a methodology, a framework for the aging studies. The questions what you are asking is actually not answered in this research report.
Hope it answers your question. So, do anyone have any other questions? I can't see any other questions. So we can move to the next. Yes. So next presentation is multi. The topic was multi-omic underpinnings, epigenetic aging, and human longevity. It will be presented by Dr. Vijay.